Welcome to the session on non-communicable diseases. In this session, we have two eminent speakers, and it is my pleasure to introduce our first speaker, Professor Cheong Sok Ching. Prof. Cheong leads the Translational Cancer Biology and Digital Health Research units at Cancer Research Malaysia. The team is involved in numerous areas of cancer research, which include the development of immunotherapy and the use of CRISPR-Cas9 essential screens to identify novel targets for head and neck cancer. Her work has received several national as well as international scientific awards. Professor Cheong holds several prestigious fellowships and is the current Dr. Siti Hasma Muhammad Ali Professorial Chair. The talk this afternoon is on targeting signaling pathways in oral squamous cell carcinoma. Prof. Cheong, the floor is yours.
Thanks a lot, Professor Umarani. Okay, I'm just going to share my slides now. Do you see my slides?
Not yet. Yes, now yes.
Let me move this away. Good afternoon, everybody. First of all, let me just thank the organizers for this kind invitation and thank Professor Umarani for the kind introduction. This afternoon, I would like to talk about a particular cancer, which is very much an Asian cancer called oral squamous cell carcinoma, or OSCC, which basically is a disease in the oral cavity. I will talk particularly about the gaps in the therapeutic options for this disease and what are the key signaling pathways for oral cancer, and some of the techniques we are using in order to identify new targets to make new drugs, particularly using the CRISPR-Cas9 technology to identify what are the vulnerabilities of oral cancer. During my presentation, I would focus on talking about building and utilizing some of the resources that are available, mainly because a lot of us who are doing research in the developing world will find it quite difficult to do expensive experiments, and therefore I would like to point out some of the resources that we can use whilst we are doing our research that are in the public domain. So oral cancer, as I mentioned, is cancer of the oral cavity, whereby cancers can occur within the soft tissues within the mouth, such as on the tongue, the floor of the mouth, the cheek, and the palate. This is very much an Asian disease. As you can see here, the darker the blue color shows you where the disease is at its highest. So therefore, you can see that within our region, which is Asia and Asia Pacific, the blue is the darkest. So this is where the disease is at the highest. Most deaths also occur in the South and Southeast Asian region. So in a nutshell, Asians make up 66% of the world's oral cancer cases and up to about 75% of all oral cancer deaths in the world. So this is the reason why we are focused on this particular disease, as we need to really understand the disease as it happens in our region so that we can come up with better therapeutic options. The mainstays for oral cancer treatment is surgery, radiotherapy, and chemotherapy, just like many other different cancers. But because the cancers usually are diagnosed at a late stage, so about 65% of oral cancer, despite easy visualization, still comes at stage 3 and 4, where the disease is very extensive. Therefore, usually there will be a need for the combination of surgery, radiotherapy, and chemotherapy. However, up to 2/3 of oral cancer patients do not respond to chemotherapy, and therefore their options remain very low. And there is a form of targeted therapy called cetuximab, which targets the epidermal growth factor receptor protein that has been approved more than 10 years ago. Beyond that, more recently, immunotherapy in the form of checkpoint inhibitors such as pembrolizumab and nivolumab are currently approved for the treatment of oral cancer. This graph shows you the number of targeted therapies that target a particular protein in the cancer cell against the cancers. So you can see that cancers on the top, such as leukemia, lymphoma, and breast cancer, have had a lot of advancements in terms of the development of targeted therapy. However, comparatively, head and neck cancer, which is where oral cancer falls under, is still lagging behind. This is very important because if you look at this graph, which depicts the increase in survival over the last few years, shows that the cancers where there are huge development in terms of the number of targeted therapies have increased survival, whereas those that are lagging behind, the survival has not improved significantly. So in order for us to be able to improve survival, one approach is to ensure that we understand the disease so that more therapies can be developed. Many years ago, it has been a long-standing knowledge that gene expression patterns of the tumor can actually predict whether the tumor is sensitive to a particular drug. These are very nice studies done from the Wellcome Sanger Institute called the Genomics Drug Sensitivity in Cancer, or GDSC in short. What this study have done is to take many different cancer cell lines from different kinds of cancers and treated them with hundreds of different cancer drugs. And you can see that certain cancer cell lines, such as the ones depicted here in the red box, are sensitive to a drug called dasatinib. And this sensitivity is accompanied by specific gene expression patterns whereby this panel of genes here are upregulated. If this panel of genes are downregulated, these cancer cells will not be sensitive to this particular drug. And a second case study using the heat shock protein 90 inhibitor is also shown, whereby the sensitive cells overexpress a panel of genes and downregulate a different panel of genes, which is opposing to those cells that are resistant. This tells us that the inherent gene expression pattern can predict whether a cell line or a tumor is going to be responsive to a particular drug. So this tells us that we may be able to put in a gene expression profile of a cancer cell or a tumor into a search tool, such as what we are familiarized with Google, whereby this search tool would then tell you what drugs this particular cancer could respond to. So this is exactly what we did. We built a Google-like tool whereby we can query the gene expression profile of a cell line and predict what drugs this particular cells would respond to. First of all, we have to build the database, which is similar to Google. What we did was we took publicly available data from the GDSC, as I mentioned just now, and built this particular database called Design. In Design now, each drug is tagged to a particular gene expression profile. So therefore, when we query a gene expression profile of cancer cells, what the search tool does is determine the similarity of the gene expression of your query to the gene expression in the database to fish out the drugs which would be most efficacious in the particular tumor. And these inhibitors will then be ranked based on sensitivity. So the most sensitive, the drug that is most efficacious would be on the top versus the ones basically will not work in their particular cancer. So this is how Design looks like. This is a live web page. You can put in the gene expression of your query tumor, the upregulated genes and the downregulated genes. And we've done that particularly with a panel of cell lines that we derive from Malaysian oral cancer patients, whereby we determine the up and down regulated genes, query in the design database, and the output of the drugs based on the most efficacious on the top to the least efficacious at the bottom. So we identified several drugs, and I'm only going to talk about two particular case studies. So first one I'm going to talk about is targeting the epidermal growth factor receptor, as I mentioned. And you can see that there are several drugs here targeting the epidermal growth factor receptor. What we then did was we tested this in the cell line panel that we've derived from our patients. Before I describe the experiment, let me just talk a little bit about the epidermal growth factor receptor, or called EGFR. So EGFR is overexpressed in more than 90% of oral squamous cell carcinoma, and EGFR signals down to many important cancer pathways, one of which is the MEK-ERK pathway, and the second one is the PI3 kinase pathway. The EGFR and the PI3 kinase pathway and the MEK kinase pathway are very important pathways that are essential for the survival of oral squamous cell carcinoma. And several small molecule inhibitors targeting the epidermal growth factor receptor, such as afatinib and erlotinib, has already been approved for many different other cancers, such as lung cancer and colorectal cancer, but not approved yet for oral cancer. So this is what we wanted to do. We wanted to test whether erlotinib and afatinib could be repurposed for oral cancer use. These are some of the experiments, and I'm just going to summarize this, and you can look at the details in the published paper. The first drug I want to talk about is erlotinib. Erlotinib is the second generation small molecule inhibitor targeting the epidermal growth factor receptor. This is basically just a cytotoxic assay, and you can see that the majority of the oral cancer cell lines are very sensitive to the drug, shown by a very low IC50. But there is also a panel of cell lines that are resistant to this drug. So one might think that, okay, this drug may not be very interesting because not all the patients are going to respond. On the contrary, when we are looking for drugs to target a particular cancer, this is the profile we actually like. We like what we call the binomial distribution, because we like cell lines that are sensitive and cell lines that are very, very resistant. Because this enables us to try and understand how the drug works, whether there's a biomarker to select the best patients who should be receiving this drug, and what are the mechanisms of action for this particular drug, and what are the actions for cell lines developing resistance to this drug. So it turns out that if we compare the genomics, that means mutations and polymorphisms, between the sensitive line and the resistant line, we found a particular polymorphism in a gene called the periplankin at this particular position 4717, whereby those cells with a GC or GG genotype are very sensitive, whereas you can see the ones that are resistant harbor the CC genotype. And we proved that this is a causative event by engineering this particular polymorphism in cell lines that were sensitive. So in cell lines that were sensitive, by giving them this CC genotype, they became more resistant, depicted by the higher IC50, and this is shown by two cell lines. We also tried to make the cells that are resistant here to be sensitive by engineering them to have the GG or GC phenotype. So you can see here for cell lines that are resistant, by engineering this GG genotype, you can see the IC50 goes down, showing that this particular polymorphism is really important in modulating the response to drug. And we postulate that periplankin is actually a platform protein trying to bring together many different other signaling proteins within the EGFR signaling pathway, and therefore by changing a polymorphism changes the protein conformation and its interaction with the partners within the signaling pathway. So this tells us that perhaps if we choose to bring erlotinib into the clinic, then periplankin could perhaps be a biomarker for this drug. We also studied a second EGFR inhibitor, and this EGFR inhibitor not only inhibits EGFR, but other family members such as HER2 and HER3. By treating the cell lines with afatinib, the cell lines had very, very low IC50 in the sub-nanomolar levels. So we were quite excited with this drug. And you can see by this Western blot that when we treat the cell lines with this particular drug, all the signaling pathways are turned off, as shown by the phosphor-EGFR, phosphor-HER2, phosphor-HER3, they're all gone, which means that the drug is targeting its particular protein and shut down the signaling pathway. We also demonstrated in mouse models that the oral squamous cell carcinoma treated with afatinib is exquisitely sensitive. You can see that the tumors are very small. However, as we prolong the treatment with afatinib, we can see a particular protein being upregulated, and this is basically the protein here called MEK. So we know that prolonged treatment with afatinib causes MEK to upregulate, and therefore this could be a resistant mechanism towards afatinib. What we then did was to combine two drugs targeting both upstream, the EGFR, and also MEK by using afatinib and also trametinib. And you can see that the combination almost eliminates the tumor. This tells us that by understanding how the mechanism of the drug works and what are the potential resistant mechanism, we can then think about combination of drugs in order for us to ensure that the tumor doesn't escape. The second area I want to talk about is an important protein, which is a cell cycle protein called CDK4. And the CDK4 is an important protein because it is always upregulated in OSCC. And this protein is very important to drive the cell cycle. And as we know, cancer cells, one of the most important phenotype of a cancer cell is the ability to proliferate non-stop. And CDK4, together with its partner cyclin D, which is also always amplified in oral squamous cell carcinoma, drives this process. CDK4 actually phosphorylates, it's a kinase, phosphorylates the retinoblastoma or RB protein, thereby releasing this particular transcription factor. And when this transcription factor is released, cell cycle progresses. But when the cells are treated with a particular drug called palbociclib, which inhibits the kinase activity of CDK4, CDK4 no longer phosphorylates RB, and RB continues to bind to E2F, which is the transcription factor, halting the cell cycle. So these are again similar experiments whereby we treated the cells with palbociclib, and you can again see we have two populations. One population very sensitive to palbociclib, and the second population not sensitive to palbociclib. And looking at the cell lines in the mouse, which are sensitive to palbociclib, you can see that the treatment with palbociclib almost eliminates the entire tumor. But what we were interested in is how to make these cells which are resistant to be sensitive to palbociclib. We found out that the difference between a resistant and sensitive line is the presence of a mutation or a hotspot mutation in PIK3CA, which is an important component of the PI3 kinase signaling pathway. You can see that if you have a mutation, the IC50 is really high. And if you have no mutation, then you become more sensitive to the drug. And again, we did a lot of genetically engineering the cells in order to engineer this particular mutation into the cell, which is in blue. You can see that the IC50 compared to the wild type cells are always increased. We also look at signaling pathways. Every time we look at signaling pathways, you will see a Western blot. You can see that if you have the mutation shown by the plus sign here, that the phosphorylation of RB doesn't go down completely, telling you that RB now releases E2F and the cell cycle continues to move. What we then found out was when we have a PI3 kinase mutation in the cell upregulates an alternative CDK, which is CDK2 and cyclin E, allowing the cells to proliferate continuously. So now that we understand how these cells escape palbociclib by having a PI3 kinase pathway mutation, what do we do about it? By understanding this, we know that the PIK3CA pathway is turned on. Therefore, we combine again with another drug that shuts down the PI3 kinase mTOR pathway. And again, you can see that with palbociclib, the tumor doesn't really resolve because this is a resistant line. And with the PI3 kinase pathway inhibitor, again, the tumor doesn't resolve, but in combination, the treatment is better, helping the tumor not to escape treatment by a single agent. So we now know a lot about signaling pathways in oral squamous cell carcinoma. For example, I talked about the mitogenic pathway in the form of epidermal growth factor receptor signaling and also CDK4. But now knowing these pathways is one thing, and secondly, knowing which component in this pathway to target is another thing. So we started looking at a different approach by asking the question, which of these proteins, when deleted, causes the cancer cell to die? And we have at our disposal the CRISPR-Cas9 technology, whereby we can actually use genetic scissors to cut out a particular gene one by one in the cell. So we utilize again our large panel of Asian cancer cell lines, which we derived more than 10 years ago, and this is called the ORL series. These ORL series are always used in our laboratory because they represent the kind of mutational status that we often see in our cancer patients. And we did the CRISPR-Cas9 screens by first engineering the Cas9 protein, which is the genetic scissors, and then we put in the sgRNA. And what we were looking for are cells that are missing because the gene that is deleted causes the cell to die. This is what we call a negative screen. We harvest all these cells and then we do sequencing. And again, as I want to emphasize, what we're looking for is the missing gene. Using a pipeline to analyze this, we identified 918 genes that are critical for the survival of oral squamous cell carcinoma. And using an algorithm we derived, we ranked all these genes. And we are quite happy to see things that we were targeting before these screens were there, such as the epidermal growth factor receptor, which is an important gene for OSCC, and also CDK6. And I talked about this in my previous slide. So these genes now can be grouped into different kinds of function. For example, many of them are transcription factors, and many of them are also protein kinases, which is very targetable. We started to bin the 918 genes to prioritize which ones we will study and which ones are really difficult to study. And they can be grouped into 3 groups, which is also talked about in our paper last year. The first group that we looked at is the genes where there are already drugs, such as the epidermal growth factor receptor and mTOR and other genes where drugs have already been approved for other cancers. The second group are where we are really focusing right now. These are genes where they are targetable but may not really have drugs yet. And the third group are proteins where not much is known about them and a lot more work has to be put in before we understand the function of these proteins. So I'm going to kind of stop there and talk a little bit about what does that mean to us in terms of knowing what are the critical genes. So by knowing which are the critical genes, we begin to think about how do we target these critical genes, because these genes, when targeted, will cause the cancer to die. If you're not interested in oral squamous cell carcinoma, but interested in other cancers, that's fine. And I want to highlight 2 very important resources that we are now contributing to, which is called the Cancer Dependency Map and also Project SCORE. The Cancer Dependency Map comes out of the Broad Institute in the US, and the Project SCORE comes out with our collaborators at the Wellcome Sanger Institute. And you can play around with the cancer that you are interested in to identify the genes that are most important in that cancer. In addition, these tools are very powerful because you can start to think about whether there are drugs targeting those gene dependencies. I just wanted to highlight that because we were able to make cell lines from our patients, and oral squamous cell carcinoma is an understudied cancer in the West, we were able to collaborate with the Sanger Institute to contribute to 1/3 of the currently available global data for oral squamous cell carcinoma. The other 2/3 are contributed, as I mentioned, by the Cancer Dependency Map from Broad and also our collaborators from the Sanger Institute. Before I end my talk, I want to talk a little bit about a new project that we are doing together with the Sanger Institute. I started my talk talking about the genomics of drug sensitivity in cancer, whereby there are 140 drugs over 700 cell lines. The database has improved since 2012, where there are now 400 drugs or more than 400 drugs treated on more than 1,000 different cancer cell lines, not only oral cancer, but many different cancer cell lines as depicted here. And together with Sanger, we have increased these models significantly for head and neck squamous cell carcinoma, whereby before we did our experiments, there were only 43 cell lines, and head and neck cancer was ranked number 7. Whereas with our data now, we have now bumped head and neck cancer to the third most largest database in the GDSC. And we've also added now nasopharyngeal cancer, which is an important head and neck cancer in our region. This would hopefully now enable us to combine different kinds of data. So for example, gene essentiality data I talked about in our CRISPR screen. With this particular data together with Sanger, we were able to now combine gene essentiality to ask the question, if a particular gene is essential, what drugs will these cancer cells respond to? And use bioinformatics analysis to identify the top drug candidates with robust gene biomarker. And where we hope to achieve is to improve the number of targeted drugs for head and neck cancer in the future so that we can improve the number of options for our patients and hopefully drive the improvement of cancer survival. Before I end, I just want to acknowledge the key people for whom the work I've described here. Design was done by my PhD student who has now graduated, Dr. Bernard Lee, together with our collaborator, Dr. Ik Chun Tan, who is a fellow Malaysian from the Moffitt Cancer Center in Florida, and also a very talented statistician from University Malaya, Associate Professor Dr. Kang. The CRISPR work was done by this team led by Dr. Annie Chai and our 2 research associates, Shiman and Pei San. Initially started with Dr. Alton McDermott, who is now in AstraZeneca, and further on with Dr. Matthew Garnett, who's also helping us take forward the GDSC work. And other collaborators who, for the longest time, has helped us more than 10 years ago establish cell lines here at Cancer Research Malaysia, and our surgeons who diligently help us collect tissue in order to make these lines viable. So I would like to thank our patients for contributing to the research. Even at their darkest hour, they found the strength to contribute, and they understand the importance of doing research. And importantly, I would like to thank our funders, for which played a huge role in order for us to do research here in Malaysia. Thank you very much for your attention. I would be happy to take any questions if there are any.
Thank you for an excellent talk, Prof. Cheong. Due to time constraint, I'm afraid we will not be able to take any questions. However, I'm sure the audience are waiting and the participants are waiting to ask you lots of questions. So I hope you will kindly answer the questions in the chat box.
Sure. Yeah, please put your questions
in the
chat
much.
box.
And I wish you all the best for all the wonderful work that you're doing. Congratulations.
Thank you, Prof. Uma. It's really nice to see you.
Yes.